To some extent, we now know the properties of an antigen, but it is also vital to understand how this antigen binds with antibodies to bring about some amazing reactions within our bodies. It is also crucial to understand the differences and properties for each of these antibodies and antigens interactions. Many of us get used with these two terms, and one can be mistakenly being thought of as another. To get rid of this confusion, in this session, we bring in a clear comparison of both these terms, the antigen-antibody reaction, types of antigen-antibody reactions, and properties of antigen-antibody reaction. Antigens are elements that provoke an immune response. They are the final objective of the immune system. Antibodies are merely proteins that are secreted as an outcome of the antigen triggered immune response. In brief, antigen cause the disease and antibodies cure it. So, in this session, let us study in detail about these two substances which put up a fight within our bodies. By a combination of the hydrogen bonds, van der Waals forces, electrostatic interactions, and the hydrophobic interactions, antigen-antibody interactions are generally non-covalent and reversible in nature. Affinity and avidity are always mentioned besides specificity and cross-reactivity when identifying or explaining the strength of an antigen-antibody complex. Let us now see the antibody affinity. The strength of the interaction in between an epitope and an antibody's antigen binding site is measured by affinity. The same basic thermodynamic principles that rules any reversible biomolecular interaction also defines the affinity. Hence, Ka is equal to Ab minus Ag divided by Ab multiplied by Ag, where Ka denotes the affinity constant, Ab is the molar concentration of the unoccupied binding sites on the antibody site, Ag is the molar concentration of the unoccupied binding sites on the antigen site, and Ab minus Ag is the antibody minus antigen complex molar concentration. The Ka explains how much the antibody antigen complex remains at the point when the equilibrium is reached. The time taken for this to happen always depends on the rate of diffusion and is same and similar for every antibody. However, in a shorter period to a larger amount of antigen, high affinity antibodies will bind than low affinity antibodies. For antibodies, therefore, Ka can vary widely, that is the range below 105 per mole and above 1012 per mole. This can be influenced by the factors including buffer composition, temperature and pH. Monoclonal antibodies are homogeneous and selective for one single epitope, and the affinity can be accurately measured. Polyclonal antibodies are always heterogeneous and will have a mixture of antibodies of different affinities, and they can recognize several epitopes. Thus, only an average affinity can be measured and determined. Coming to antibody avidity, The overall strength of an antibody antigen complex is measured by avidity. On three major parameters, the avidity depends. For the epitope affinity of the antibody and antigen's valency, the interacting parts structural arrangement. All antibodies are said to be multivalent. For example, immunoglobulin G are always bivalent and IgMs are always decavalent. When the number of antigen binding sites are higher, that is more the immunoglobulin valencies, the affinity to bind to the antigen is also higher. Similarly, multivalency can be demonstrated by antigens since they can bind to more than one antibody molecule. The stabilization of an antibody and an antigen is aided by the multimeric interactions between them. The stability of an antibody antigen complex can be led by the structural arrangement of an antibody and an antigen. There is an opportunity that the antibody antigen complex is provided by an immobilized antigen to form, that is a high local concentration of the available epitopes than a free antigen in the solution over same time period. The chances of a bivalent interaction are higher once the antibody's first antigen binding arm attaches to an antigen on a support. This principle is used by many immunoassays like Western blotting and ELISA technique. The large stable structures will be formed when an antigen is mixed up with a polyclonal antibody. The formation happens due to multivalent interactions. This is because to several antibodies an antigen may bind, each recognizing a different epitope. Polyclonal antibodies are hence ideal for the immune precipitation experiments. Coming to the specificity, the ability of an individual's antibody combining site that reacts with only one antigenic determinant is referred to as the specificity of an antigen-antibody reaction. In nature, antigen-antibody reactions are highly specific, that is, one antibody can react with its own antigen molecule. A strong antigen-antibody interaction usually depends on a very close fit between the antigen molecule and antibody molecule that requires high degree of specificity. The high specificity and affinity is the most striking features of the antigen-antibody interaction. But in some instances, antibody evoked by one antigen molecule can cross-react with an unrelated antigen molecule. This kind of reaction is known as cross-reactivity. Now let us see about cross-reactivity. Cross-reactivity is often observed in polysaccharide antigens that contain similar oligosaccharide residues. On red blood cells, A, B, O blood group antigens that are glycoproteins are expressed. Distinct differences in the terminal residues of the sugar molecules attached to the surface of these proteins differentiate the A and B blood group antigens. Serum antibodies to the missing antigens will be present if an individual is lacking one or both of these antigens. Thus, serum group of O and group B have anti-A antibodies and group O and A have anti-B antibodies. Anti-A and anti-B are believed to be not present in group AB individuals because group AB individuals express both antigens on their red cells. Cross-reactivity is also demonstrated by some vaccines. Now we will discuss about the types of antigen-antibody reactions and also their properties. Let us begin with the precipitation reactions. When a soluble antibody reacts with a soluble antigen to present an insoluble product or a precipitate, then the reaction is called as precipitation reactions and it produces the precipitins. Soluble antigen that stimulates the formation of a specific precipitin is called precipitinogen. By changing the concentration of the antigen, there is a marked effect on the precipitation reaction. A quantitative precipitation reaction can be executed by placing a constant quantity of antibody in a series of tubes and further adding increasing quantity of antigens to the tubes. By plotting the amount of precipitate against the increasing antigen concentrations will yield a precipitation curve. A precipitation curve for a scheme of antigen antibody shows three zones. The first zone is the zone of antibody that is in excess or called as pro zone, in which the concentration of the antigen is very low and that of the antibody is comparatively high. The second zone is called as the equivalency zone and also the zone of maximal precipitation, in which antigen and antibody form large insoluble complexes, and in the supernatant there will be no antigen or antibody present. In the third zone, the antigen will be present in excess amount or also called as post zone, in which the concentration of the antigen is very high. Let us see the types of precipitation reactions. In solution phase and in gel phase, precipitation reaction will occur, where a precipitate is formed by antigen antibody. In fluid, as the precipitation reaction happens, visible precipitation occurs in the region of equivalency and no visible precipitate is formed in the regions of antibody or antigen excess in the gel phase. Let us see the fluid phase precipitation. This method is a double diffusion method, where over an antibody solution, an antigen solution is layered in a capillary tube. Towards each other, both the antigen and antibody will diffuse, and at the boundary between them, when antibody identifies an antigen, precipitate forms. Depending on the concentration of both the antigen and antibody, the precipitate is formed. Using this method, unknown antigen or unknown antibody can be identified. The next one is gel phase precipitation. Instead of solution in gel phase precipitation, a gel is used as a semi-solid medium. There are pores present in gel that allows the movement of the molecules. The gel is called the agarose gel. When an antibody is incorporated into the agar, an antigen diffuses into the antibody containing matrix, or when antigen and antibody diffuse towards one another in agar, a visible line of precipitation is formed, unlike a precipitation curve in fluids. To determine relative concentrations of antibodies or antigens, two types of immunodiffusion reactions can be used. They are double immunodiffusion and radial immunodiffusion. Let us now pass on to the agglutination and the types of agglutination reactions. The agglutination reactions can be classified into three types. The three reactions are quantitative agglutination or bacterial agglutination, qualitative agglutination or heme agglutination, and passive agglutination. Let us get to know about each one of them in detail. Let us first see what is quantitative agglutination or bacterial agglutination is. Using agglutination, one can determine whether a patient had or has a bacterial infection. Quantitative agglutination test is the other name of agglutination reaction, where here the measurement of level of antibodies to particulate antigens is being done. By bacterial agglutination reactions, the presence of serum antibodies in a specific person, particular for surface antigens on bacterial cells, can be identified. If a patient is believed to have typhoid fever, The patient's serum is then mixed with a culture of Salmonella typhi. If an agglutination reaction occurs and if it is shows the clumping of the bacteria, the patient is believed to either has or had an S. typhi infection. Since certain antibodies can remain in a patient's blood for many years, even after the patient has recovered from the infection, a positive reaction will not mean that the patient presently has the infection. To find out whether a patient is currently suffering from typhoid fever, the amount of the antibodies will be identified at the attack of illness and 2 weeks later. In an array of tubes, the serum from the suspected patient is diluted serially, to which the bacteria is added. In the last tube, visible agglutination can be seen, which will then reflect the serum antibody titer of the suspected patient. The reciprocal of the greatest serum dilution that evokes a positive agglutination reaction is defined as the agglutination titer. Naturally, in an individual, when the titer is higher, the response of the antibodies will also be higher to the disease. The patient at present suffering from the suspected typhoid fever will show a substantial rise in the agglutination titer to Salmonella typhi. Agglutination reactions also aid to type bacteria. Qualitative agglutination or heme agglutination. To assay for the presence of an antigen or an antibody in a qualitative manner, qualitative agglutination can be used. With the particulate antigen, the antibody is mixed and a positive test is pointed by the agglutination of the particulate antigen. Heme agglutination is a particular form of agglutination that requires type of red blood cells. The blood group ABO antigens are always intrinsic red blood cell antigens, in which A and B signs refers to the proteins on the surface of red blood cells. Individuals having A antigen are indicated as blood type A. Likewise, those expressing B antigens are indicated as blood type B. Individuals expressing both A and B antigens are indicated as blood type AB, and those not expressing either A antigen or B antigen are indicated as blood type O. Blood type can be decided by using antibodies that bind to the A blood group or B blood group in a sample of blood. For the ABO antigens, red blood cells are mixed on a slide with the antisera to the A blood group or B blood group antigens. If the antigen is present on cells, they agglutinate by forming a visible clump or bunch on the slide. For example, if agglutination occurs when antibodies bind the A blood group that are added, the blood is either type A or type AB. To determine if it is between type A and type AB, antibodies that binds the B group are added, and if the agglutination does not occur, the blood type is determined as type A. In blood grouping, the patient's serum is tested against red blood cells of known blood groups, and also the patient's RBCs are tested against known serum types. By this way, patient's blood group is affirmed from both serum and RBCs. The next one is passive agglutination. This is similar to agglutination reaction, but is performed with soluble antigens. This is the agglutination of particles coated with soluble antigen and antiserum specific for the adsorbed antigen. Passive heme agglutination is a sort of passive agglutination in which the erythrocytes are normally altered by the mild treatment with tannic acid or chromium chloride are then used to adsorb soluble antigen on their surface. Into the mica titer plate wells, the serum containing antibodies are serially diluted and the antigen coated red blood cells are further added to each well. By the size of the feature spread pattern of the agglutinated red blood cells, agglutination is assayed on the bottom of the well. With tanned erythrocytes or synthetic particles, passive agglutination can be performed. The advantages of consistency, uniformity and stability are offered by the synthetic beads. The linking of the latex particle together by the antibody molecules is the initial step. The latex particle attaches specifically to the antigenic determinants on the surface of the particle. Large lattices forms through these crosslinks and the large lattices sediments readily due to the large size of bunches and are visible to an unaided eye within minutes. By plotting the agglutinant concentration that gives a bell-shaped curve, the degree of agglutination can be determined. Using the latex particles, the antigen-antibody complexes can be magnified. To conclude, in this session, we have discussed about antigen-antibody reaction, properties of antigen-antibody reaction and the types of antigen-antibody reactions. We have also discussed about agglutination reactions. The antigen and the antibodies combine precisely with each other. This interaction between them is called antigen-antibody reaction. Precipitation reactions, fluid phase precipitation, gel phase precipitation are the types of antigen-antibody reactions. The agglutination reactions can be classified into three types. They are qualitative agglutination, quantitative agglutination or bacterial agglutination, and passive agglutination or heme agglutination.